Hey everyone, how's it going? It's a nice night, so I came down here to the pond, talk to you a little bit. I want to talk to you a little bit about genetic control, transfer, and screening. So basically, just to take what we've learned now about DNA and RNA and proteins and how we can use those and how we are using those to expand our knowledge about medicine and about lots of things. Agriculture, as I said, 21st century is going to be the century of biology. And the fact that we've learned so much about DNAs that we're learning so much more, and our understanding of biology is growing at an exponential rate. And so there's not really a facet of our society that's not going to be touched by this at some point. And so a lot of these just interesting examples, at least things that I found interesting. And so we're going to talk briefly about controlling genes, then genetic transfer or GMOs, genetically modified organisms, and then finally genetic screening. So I mentioned before, it's an interesting fact that every cell has all the chromosomes and all the genes. And so whether that cell is in your big toe or your intestine or your eye, it all has all of your DNA. And so how do we get different cells? We need to be able to, how do we get different cells, and how do cells modify their behavior, quote unquote? So for example, skin cells that produce more melanin when they're exposed to sunlight. How cells that react to their environment. How do we do this? We have to be able to turn genes on and off. And I put those in quotes, but they're really not really in quotes. It's a very appropriate metaphor here. Most genes are off most of the time, which means they're not being transcribed. Or they're being transcribed, but then it's not being, the protein is not being translated. And so if a gene is not being transcribed, or it's being transcribed, but the protein's not being translated, then that gene, is off. It doesn't exist, it doesn't do anything. And so how does your body do this? Well, that's a big question. Well, there's many ways that we can control genes or turn them on and turn them off. And if you take genetics, you'll get into this in quite a bit of detail. I'm going to talk about the more common ways to control genes. For example, we can control the binding of RNA polymerase. And so you recall at the start of every gene, you've got a promoter. And that promoter is a region where RNA polymerase binds and then transcribes the gene. Well, if we can control how well RNA polymerase binds, then we can control that gene. And so if you can cover up that promoter or do something to make RNA polymerase not attach there, then the gene won't get transcribed and it's off. Another common way to control genes is methylation of cytosine bases. And this is a methyl group is a CH3 group. So methylation is adding a CH3. And so, cytosine is just your C nucleotide. And so this is just adding a CH3 to that nucleotide. And so that's a fairly simple reaction. Usually it occurs when you've got a CG next to each other. And so if on one strand you've got a CG next to each other, of course on the other strand you're also going to have a CG next to each other. So you're going to have two C's right across from each other at a diagonal. And so if both those C's get methylated, it sort of hooks them together. And so that's what they're showing you in this figure from your book. And so the methyl group attaches to the nitrogenous base, not the sugar, but the nitrogenous base, so the inner part of the DNA molecule. And so that hooks these cytosine bases together, and that stops RNA polymerase, stops all the enzymes from being able to transcribe that gene. So it stops the gene from being able to being spread apart or whatever. It blocks transcription by physically altering the shape of DNA. And so that's a very easy, quick and reversible way to shut off a gene. So you could very easily just remove those methyl groups. Now that gene can be transcribed. Like I said, there's lots of other ways we can control genes. Differential mRNA processing. You remember that we have to, take out introns and splice together exons. And if you don't do that right away, then the gene won't translate properly. You can control translation in other ways. There's lots of things you can do to make sure that that gene is or is not turned into a protein. And since most of your genes are off most of the time, control of those genes can be very important. So, like I said, that's a 10,000-foot view, but it's just important for you to think about that we can turn genes on and turn genes off. Okay, next thing I want to talk about, just some interesting stories about genetic transfer. Moving genes among species and creating genetically modified organisms. And so GMOs, or genetically modified organisms, that's a buzzword that you hear a lot. You certainly hear it a lot in the grocery store, and you see a lot of your foods are, no GMOs and things like that. And so I think it's important that we as biologists understand what this means. And another word for a GMO is a transgenic organism, meaning that we take a gene from one species and put it in another species, and it gets transcribed and translated. And I've given you some examples of this earlier, but remember, this works because the genetic code is universal. And so that gene, the sequence of that gene and the codons that are made in the RNA are going to make the same sequence of amino acids. So let me give you some examples of this. For example, we could take a gene from a mammal, put it into a bacterium, and it will function. Now think about that. You could not get two more distantly related organisms. You could not get two more different organisms than a mammal and a bacterium. However, you take the gene from one, you put it in the other, and it works. And so a good example of this is bovine growth hormone. Bovine growth hormone is a gene, and it makes the bovine growth hormone protein. And which is abbreviated here BGH. And so that BGH controls milk production. And so it's been well known in the dairy industry that if you give the cows more BGH, they'll produce more milk. And so you can extract that BGH from living cows, but of course that's takes a long time and it's difficult to do. It's slow and it's expensive. But if this gene makes the protein, what if we take that gene and put it into a bacterium? And that's what happened. So why would you want to put this gene in a bacterium? Well, bacteria grow very quickly and reproduce very quickly. And every time that bacterium reproduces, it makes a new copy of that BGH gene. And so all the offspring have that BGH gene. And so, every 20 minutes, every 30 minutes, you can make a new copy of that gene. And bacteria are easy to raise, and they don't eat very much. And so you can feed them the right substrate, and they will transcribe that BGH gene and then translate it into the protein. And so then the bacteria will be making this bovine protein. And since there are so many of them, and since you can feed them very cheaply, you can make a tremendous amount of this protein by having the bacteria do the work for you. Then you just harvest the protein. So that's an example of a transgenic organism. And so here's kind of the steps. I think there's a figure from your book. And the first thing they're showing you here is you find the gene and you cut it with a specific enzyme. And so there are enzymes that their whole job is to cut DNA. But a lot of these enzymes leave what are known as sticky ends. So they look for a specific sequence and they cut at that sequence, but they leave one strand a little longer than the other strand. And they do that at the same sequence on either side of this gene. Then you take the bacterial genome the bacteria gene, which again you see, is a circular, just one circular chromosome, and you cut it with the same enzyme. And so that enzyme is going to cut in the same sequence and leave the same sticky ends. So then, when you mix that bacteria and that bacterial DNA with the bovine gene, which you cut, it's got the same sticky ends, those sticky ends are going to match up, and now you've stuck that bovine gene right into the bacterial genome. And so then you just let the bacteria reproduce, and then they'll reproduce that gene every time they make a copy of themselves. And so you can make a lot of protein quickly and cheaply this way. And it's not just in agriculture. This is done to make human insulin or blood clotting factor, important proteins that we need for human diseases. You can use transgenic bacteria to produce those quickly and cheaply. And again, because the genetic code is universal. Okay, so that was a mammal gene into a bacterium. Let's go the other way. Let's take a bacterial gene and put it in something. And so an example, another agriculture example, is Bt corn. And so that's a gene from bacterium that's been spliced into corn. And now every cell of the corn has this bacterium gene. And so Bt stands for Bacillus thuringiensis. And that's just a bacterium, a species of bacterium. This bacterium has a natural protection from certain insects. And so all organisms are always competing. And so organisms evolve defenses against other organisms. And this is one defense that this bacterium has against these insects. Well, the same insects also prey on corn. And so how does the bacterium protect itself? That bacterium has a gene that produces a protein. That's what genes do. But this particular protein binds to the gut wall of these insects and breaks it down. And so the insects that ingest the bacterium, their gut wall breaks down and it kills them. So scientists cut this gene out of the bacterium, spliced it into corn. Now the corn produces that same protein. And so then those insects also like to eat corn, but the same thing happens. If they eat the corn, the protein binds to their gut wall and it kills them. And so that's another example of a GMO. Here's a good example of a genetically modified animal. So we had a genetically modified bacterium, a genetically modified plant. This big fish in the back, these fish in this picture are the same age. They're siblings actually, I think. And so the one in the back obviously is growing a lot faster, and that's a transgenic salmon. Excuse me. And so this is famous because it's the first GMO animal to be considered for sale and to get approval, actually. It's been approved. And so how is this fish genetically modified? There's a gene for a growth hormone from a Chinook salmon that got spliced into this Atlantic salmon. Those are different species. Then the promoter for that gene came from an ocean pout, which is a completely different species, and I think it's in a different family even. So an Atlantic salmon is a popular fish, and they're easy to grow in captivity, but they don't grow very quickly, they don't grow very large. If you could get them to grow larger faster, you could maybe make more money. The Chinook salmon does not do well in captivity, but it's very big. And so that's why its growth hormone is much more effective and makes fish grow faster. So by putting that Chinook salmon growth hormone in the Atlantic salmon, you can make the Atlantic salmon grow faster, but still retain the other characters of the Atlantic salmon. This promoter came from a different species, this ocean pout, which is a different fish species. Recall again what the promoter does. That's where the RNA polymerase binds and starts transcription. And we said earlier that controlling genes, one way to control genes is to control the binding of RNA polymerase. Well, some promoters work better than others. And some promoters are better at binding RNA polymerase than others. This happens to be a very good promoter. And so by putting this good promoter in front of this good growth hormone, not only can you produce this good hormone, you can produce more of it, and that's how you can get the fish to grow larger. So you get very fast growth in Atlantic salmon. Another agricultural example, transgenic strawberries. So that was moving some genes among different species of fish. Now we're going to talk about taking a gene from a fish and putting it into a strawberry plant. And so there are some Arctic fish, like the crocodile ice fish, which is a cool name, and they live in extremely cold water. And they have what are known as antifreeze proteins. So they have genes that produce these proteins, and the function of those proteins is to just act to keep from ice crystals from forming in the blood. So these fish live in the Arctic water, where the water is actually below 0 Celsius. You say, well, how can you have water that's below 0? Doesn't water freeze at 0? Yeah, but salt water has a lot of stuff dissolved in it. Remember that salt disrupts the lattice, and so you have extraordinarily cold water, and you have fish that live in this cold water. They also are in danger of having ice crystals that form in their cells. And so these antifreeze genes produce a protein that stops the ice crystals forming. Well, ice crystals forming is a problem for lots of organisms. And in this example, we're talking about strawberries. Why do strawberries care about ice forming in their cells? Well, when ice forms, of course, it expands, right? When you form that lattice, it expands. That's why if you put a can of, soda in the freezer, it'll explode. Well, the same thing will happen inside cells. And so if ice crystals are allowed to form inside cells of a strawberry, those cells are all going to burst open. And that can, possibly kill the plant, but it certainly can make the strawberries very mushy. And so if you get a hard freeze while your strawberries are still on the vine, you can lose those strawberries because all their cells burst because of the ice crystals. So by taking and putting this fish gene in the strawberries, maybe you can stop that from happening. So again, remember that when water freezes, it makes that crystal lattice. Anything that can stop that lattice from forming will stop ice from forming. So salt does this, but you can't really increase the salt content of your cells without messing up a lot of things. But you have these proteins that get in there and stop that lattice from forming. That'll work. And so if ice forms in the cells, they'll swell and burst. That's why cryogenics doesn't work. That's why you can't freeze somebody and then thaw them out later, because the ice crystals in their cells will cause all their cells to burst. This is why food gets squishy after you freeze it and thaw it a few times. And so you put something in the freezer and you thaw it, then you put it back in the freezer and you thaw it, and then you eventually eat it. The food gets real squishy because those ice crystals are breaking down the cell walls. And so the idea was, does this gene works in fish, will it work in strawberries? There's also been people trying to put this in tobacco. Same idea. It makes it more resistant to a hard frost. There are other plants that already produce their own antifreeze protein, like carrots. And the fish gene in strawberries and tobacco, last I looked, it kind of worked. It didn't work great. But by taking a plant antifreeze gene and moving it into other plants, that seems to work better. And so the intracellular environment seems more amenable to this. So anyway, that's just another cool example of a GMO or a transgenic organism. Now, one of the complaints against GMOs is that, it's quote-unquote playing God, or it's something that we shouldn't be doing, that we shouldn't be moving genes among species because that's not natural. Well, is it? Is it unnatural? Is moving genes between different species unique? No, it is not unique. And in fact, you find that genes get transferred among species all the time. And so this is not something that humans came up with. organisms have been doing this for billions of years. Now, how do we know that? Because we can sequence the DNA now. And so we can sequence the genome of all these different species, feed them into a big computer, and say, find me a match. And you can find stretches of the DNA that are base by base almost identical in these radically different species. And that's not going to happen by chance. They had to have, either a common ancestor or a different way of getting that DNA incorporated from one species to another. So let me give you some examples. Bacteria do this all the time. It's called horizontal gene transfer. And so bacteria, of course, are asexual. They reproduce just by splitting in half. They don't have sexual reproduction. But they do exchange DNA sometimes. And so they pass DNA from one bacterium to another, from one species to another. That's called horizontal gene transfer, and it's incredibly common. This is just something bacteria have been doing, again, for billions of years. There's lots of evidence that bacteria can transfer DNA to their host. So, for example, there's a species of bacterium called agrobacterium, and it's so good at transferring DNA to plants that it's used in genetic engineering. Like, how do we get the gene into the plant? We put it in agrobacterium and then infect the plant with agrobacterium, and the agrobacterium does it because that's what agrobacterium does. It does this. It's been doing this, for a long time. So it's really good at transferring DNA. Lots of examples of parasitic plants absorbing DNA from their host. So you have things like mistletoe and lots of plants that grow and parasitize other plants. And if you look at the DNA of both those species, you can see genes from one that are very, very similar to genes from the other. But they don't have a common ancestor. They're not closely related in evolutionary time. They didn't get those genes from, they're not identical by descent. But the only other way is that the parasitic plant absorbed DNA from its host. Insects transfer DNA to their host all the time. And so you can find all kinds of DNA in mammals and other organisms that matches DNA that's found in insects. So here's a study that I read about a few years ago, and it's pretty cool. It's a study of some fungi that are symbiotic with grasses, right? So symbiosis, of course, is two species that live in very close relationship to each other. And so this is a fungus that lives in a grass. And it's a mutualistic relationship. The fungus benefits because it can get nutrients and things from the plant. But the plants in this example, the grasses benefit because they get resistance to insects. And so this is like that BT corn. It's something gets produced, and so then the insects, when they feed on the grass, they ingest that protein, and the protein kills them. And so the grass get resistance from the fungus. You have the same species of grass that's not infected by the fungus, and it doesn't get the insect resistance. Okay, so the fungi have this resistance. Well, how did they get that resistance? So we look at the DNA. So if you look at the DNA of the insect resistance gene in the fungus, you find it's very similar to a gene that's found in a bacterium. And this bacterium is a symbiont with a roundworm. And we'll talk about roundworms in lab a little bit later. So here you've got a different symbiotic relationship, right? You've got this roundworm that's got this bacteria that lives in it. And so the roundworm, when it infects an insect, then the bacterium produces a protein that kills that insect. And I don't know what the advantage is here, but it's just like that BT bacterium earlier. It produces a protein that kills an insect. Here you've got a bacterium that produces a protein that kills that insect, but this bacterium lives in a roundworm, okay? So probably what happened was a roundworm with its bacterium friend infected an insect, which is what they do. Now normally, that roundworm bacterium combo will kill the insect, and maybe it did later, but before the insect got killed, it fed upon some grass, because that's what insects do, right? That grass that it fed on just happened to be infected with the fungus, okay? Well, in the process, that roundworm bacterium got into the plant, you know, in the saliva or whatever. Or it got into the fungus, or somehow that roundworm and bacterium got into the plant as the insect fed on the plant. And then over time, the gene from that bacterium, which lived in the roundworm, got into the fungus, which lives in the grass. You following me? You say, well, that sounds really far-fetched. Well, how else do you explain it? How else do you explain that you have a stretch of DNA in this fungus that is nearly identical to a stretch of DNA that's in this bacterium? They do not have a recent common ancestor. It's not identical by descent. And all these organisms all interact with each other. So the most logical explanation is that somehow that DNA got transferred when that insect fed on the plant. So originally the fungus was probably a parasite of the grass. And so the grass did not benefit by the fungus being there. But once that fungus absorbed that insect resistance gene from the bacterium, now the grass does have an advantage to being infected by the fungus. Now you go from a parasitic relationship to a mutualistic relationship. And so both species benefit and both their fitness went up. So that's pretty cool. Oh yeah, you know what else is cool? The bacterium I'm talking about, it's luminescent. It glows in the dark like a firefly, like lots of bacteria do. Now come on, tell me that's not cool. Tell me that is not cool. And that is a long, convoluted, complicated story. But the point of the story is that we see evidence of this everywhere. Genes being transferred from one species to another. We did not invent this. Humans did not invent this. It's been going on forever. Okay. Well, so I'm recording this during summer 2020 and during the COVID pandemic. And so viruses are on everybody's mind. Let's talk about viruses and how they're related to this topic. A virus is an excellent way to transfer DNA among species, and we see lots of evidence of this. So again, what is a virus? You're probably familiar with this by now, but it's a piece of DNA or RNA with a protein coat, and that's it. And so they're not really considered living organisms because they don't really harvest energy. They don't reproduce on their own. But what they do is they infect a host. And so the protein coat is usually the key to infecting the host. So there's something on that protein coat that allows the virus to bind to the cells of the host and then get incorporated into the cell. And so once that happens, then you've got that virus DNA or RNA, and it somehow it inserts itself into the host DNA. And so that's common of most viruses. And so then it uses the host to make copies of itself. So it hijacks the host's cellular mechanisms. And instead of making, replicating your own DNA and transcribing and translating your own DNA, the virus takes over, and all that cell does is make copies of the virus and make virus proteins and repackage the new copies of the virus in the virus proteins. And eventually the cell bursts because it's made a gazillion copies of this virus, and then the viruses go and attach to other cells and do it again and do it again. And so this is how a virus spreads, but it incorporates itself into the host DNA. And so here's just a typical schematic of a flu virus, and those proteins that stick on the surface, like I said, those are key to binding to the host cells. I think it's got something to do with why COVID is so infectious, is that there's a mutation, and now it's very good at binding to epithelial cells in the respiratory tract. I think we're going to watch a video later about this. Anyway, you see it's just a protein coat with some DNA inside. Sometimes it has RNA inside. Well, these viruses can accidentally carry a gene and move it to a new host. So the virus inserts itself wherever, and maybe they have a place they target, but they don't have a lot of control. So the virus is going to insert itself just somewhere in the gene. It can insert itself somewhere in the DNA, excuse me. It can insert itself like right next to a gene. And then it starts to make copies of itself. But if it's right next to a gene, it's possible to make copies of that gene too, or to treat that gene as part of the viral DNA. And so now when you make a copy, it's not just the virus DNA, but it's the virus DNA plus this gene it sat next to. And then that gets packaged up and goes and infects another organism. And now you're putting not just the virus DNA into the new host, but also that new gene. And so you can move a gene from one organism to another. And so you can get it to go from one species to another, right? Because that's, again, this COVID probably started in another species. A lot of these viruses start where you have humans in contact with large numbers of other species, and you get a mutation that makes the virus jump from one species to another. And if it's bringing a gene with it, now you can have a gene transfer among species. Another way that we've seen viruses affect the DNA. The virus can, it just sticks itself wherever. And we've seen examples where the virus has stuck itself in a promoter and changed that promoter enough to make it a better promoter. And a good example of this is the amylase gene, which you all have, okay? So if you sequence human amylase gene and you look at the promoter, you see a sequence in that promoter that is identical to a retrovirus. So a retrovirus is one of those viruses that has RNA, and so then the RNA has to get turned into DNA, and then the DNA gets incorporated, and you'll learn all that in virology or immunology or other classes. I'm not an expert on this, but I knew a retrovirus is just the same thing, only it's RNA. At any rate, if you look at our DNA, you can see in this amylase gene, in the promoter region, you can see sequences that are identical to another retrovirus, which means that somewhere in our past, some human got infected with this retrovirus. It just so happened to stick itself in that gene, and then that got passed on to all the other ancestors. Well, this is really important to us because it's actually very important to our evolution, because amylase gives us the ability to digest carbohydrates, and gives us, or at least a better ability to digest carbohydrates. And so in our early ancestors, someone got infected by a virus, it incorporated its amylase gene, which gave them, a better promoter, a better amylase protein, a better ability to digest carbohydrate, a whole other food source. If you look in our nearest relatives, they do not have as good an amylase gene. They do not eat as much carbohydrate, but we eat a lot of carbohydrate. So that's a whole other energy source, which gives us even more energy, which allows this big brain to grow more because now you can fuel it better, and that leads to our evolution. So part of our evolution, part of the story of our evolution as humans is having this virus DNA stuck in our DNA, and it actually helped us. Tell me that's not cool. That is cool. Okay. Of course, we can take advantage of this in medicine. And so, viruses accidentally can insert genes into the DNA. Well, can we use the virus on purpose? And so an example of this is the cystic fibrosis gene. And so people who have cystic fibrosis, their gene, I forget, it's a transport gene for moving ions across a membrane, I believe. But patients who have this disorder, they can't transport salts and ions very well, if I remember right. Don't quote me on this. I can't remember the details, but I do know that it has to do with osmosis and water moving across the cells, and the end result is that you get this very thick mucus in the lungs because you don't get enough water transported to thin out the mucus, because you don't transport ions very well because of this defective gene. And so if you can incorporate a good gene into the respiratory cells, then they can function properly and you don't get that thick mucus. And so one way to do that is to get the good copy of the gene, insert it into a virus that is otherwise, benign, that's not going to make you terribly sick, infect the patient with the virus. When the virus infects their cells, it will bring that good gene with it. And then, hopefully when those cells divide, they'll keep the good gene, or you can keep infecting the cells with that. And so it's just a way to use a virus to insert a good gene for genetic therapy. Okay. So that's tons of examples of GMOs. Like I said, it's a buzzword that a lot of people, I don't think, realize the context of transgenic organisms, and don't realize that it's very common for genes to be moved among species. Certainly as biologists, it's important for you to have a deeper understanding of this, which is why I brought it up. But of course in your genetics classes, you're going to talk about this in much more detail. And these are what we would call, some would argue you can't say when something's good or bad, but these are things that we would consider beneficial to humans. Well, are there any bad examples? Again, this is a bad, saying good or bad, that kind of a value judgment isn't really appropriate, but you know what I mean, right? Like, are there any examples of, genetic engineering that we would consider bad for the human race? And, I made this slide a couple years ago, long before this COVID outbreak, but this is something that we have been talking about for a long time. Now that's the flip side of understanding this DNA and understanding these processes, is that people could use this for things like bioterrorism. And so this COVID is not a bioterrorist attack, but it shows what could happen with such an attack. And Bill and the rest of us in biology were saying this years ago, is that this is a huge looming threat and we're not prepared. And you could argue our response to the COVID breakout shows that. And maybe we caught a break. Maybe this will wake us up and we'll realize, oh, this is very serious and we need to be prepared. And, I would argue that. I would argue that we need to spend much more time worrying about things like pandemics and bioterrorism, because I think they're much more likely to happen than some of the other things we worry about. So what do we do about it? Hey, you're already doing the right thing. You're in school. You're learning more about biology. We need more biologists. We need more people in med school. Schools need more money. I can go on, but we could do a lot more to learn about this and to protect ourselves. Okay, so the last thing I want to talk about is genetic screening. Basically, another upside of understanding human DNA in the detail that we have now is that we can start to understand diseases better, and we can start to look for them, and then hopefully treat them better. And so we can start testing for a lot of diseases. So an example here is ALS, right? Lou Gehrig's disease. And so very recently, researchers sequenced DNA from people who had ALS and those who didn't. And the interesting fact here is some of the money for this research was raised by the Ice Bucket Challenge. Do you remember that? That was so long ago, like 2 years ago. It was that crazy thing on Facebook where, you dare somebody to dump a bucket of ice on their head, and if you didn't want the ice bucket, on your head, then you would donate money to this group. And so from the money they raised from the Ice Bucket Challenge, they gave it to researchers, and pretty quickly they were able to find a mutated allele in this one gene locus that ALS patients had more often than those who did not have ALS. And so it just goes to show you that, if we're given time and given money to do this research, we can do good things. So this allele is in the NEK1 gene, and a mutated form of this gene tends to show up more in patients with ALS. So what does that mean? Well, what does this gene do? And so I did a search for it, and it's been linked to several cellular functions, including cellular formation, DNA damage response, microtubule stability, neuronal morphology, and axonal polarity. So those last two things, neuronal morphology and axonal polarity, that's nerves, right? So this is a gene that is important for nerve development. ALS is a nervous disorder, and so you start to think there might be a link here, right? People who have a mutated form of this gene might get ALS at a higher rate because this gene is controlling the formation of nerves. And so I have not looked at this more recently. There's no gene therapy for this yet. You know, that's just the first step in many steps to figuring out a cure or figuring out a treatment. But it's also useful for genetic screening. And so it's the sort of thing where you could test for it, and if you find you're carrying this allele or a couple of copies of this allele, you might be at higher risk for ALS, and there might be things you can do to help delay the onslaught or delay the disease altogether. I don't know anything about this disease, but I'm just using this as an example of the advantages of knowing the DNA sequence. Here's another example of benefits of genetic screening, MTHFR, which stands for methylene tetrahydrofolate reductase. Of course. Well, you see that reductase, you see that ACE, you know it's an enzyme, right? This gene makes an enzyme that converts homocysteine to methionine or cysteine. Methionine and cysteine are amino acids, right? So you get this homocysteine needs to get broken down into these amino acids, and this enzyme does that. And so we know where the gene is exactly. It's on chromosome number 1 between those base pairs. You can see the picture down here. It's right there on the edge of the chromosome. Excuse me. And this enzyme needs folic acid as a cofactor. I think I mentioned this before, but what is a cofactor? A cofactor is a molecule that helps an enzyme hold its shape. And so again, shape is all, is everything to a protein. And so sometimes you have other molecules that just help the protein hold its shape. Lots of vitamins. So when you take a multivitamin, lots of vitamins are cofactors. An iron molecule in a hemoglobin is a cofactor. They help it hold its shape. Well, for MTHFR, it's folic acid is its cofactor. And so there's a very common mutation that changes a C to a T at base number 677 in this gene. So again, this is the advantage of being able to sequence these things. You can find out exactly where the mutation takes place. It's called the C677T allele. Now, in this example, by that one base substitution, you change the amino acids. You substitute a valine for an alanine in the polypeptide. And people who are homozygous for this C677T allele have homocysteine that builds up in their blood. Remember that this is an enzyme that breaks down homocysteine. And so if you're homozygous, if you have 2 copies of this allele, you do not break down homocysteine very well. Well, big deal. Who cares? What's the problem? The problem is that high homocysteine levels are associated with a lot of blood clotting disorders. And so if you have these 2 alleles, you might have a lot of blood clotting disorders, and especially it's important to the developing fetus. And if the mother has these alleles and this blood clotting disorders, then she can throw a lot of clots that can affect the developing fetus, and it's been linked to things like spina bifida, a lot of miscarriages, and so that's one reason why this is an interesting problem. So this allele is most common in Hispanics, next most common in Caucasians, and then Africans here in the United States, I think. So again, you'll see these genetic disorders. We know that certain groups tend to get more genetic disorders based upon where their ancestors evolved, and this is an example of that. So remember that folic acid is a cofactor. And so if you were a woman that had these alleles, you can compensate for them by taking much higher levels of folic acid. Now, if you're familiar with pregnancy, one of the things that when you're pregnant, one of the vitamins and things they put you on, the supplements they put you on when you're pregnant, is folic acid. And so I don't know for a fact, but I think probably that at some point someone realized, hey, higher doses of folic acid mean lower rates of miscarriages, and this might be one of the reasons. Because people taking folic acid that also have this allele, a couple of these alleles, this helps to compensate for that. So it's an example of why genetic screening can be important. Because if you know you've got these alleles, well, immediately you know I need extraordinarily high levels of folic acid to try and protect the developing fetus. Okay, so I kind of know a lot about this gene. I've looked at this more than others. Why do I care so much about this gene? This fella right here. This is my nephew Jake. This is an old picture. You can tell by looking at both of us. And so this is when Jake, if I had to guess, looks like he's about 2 there, right? Well, look at this picture. Do you notice anything about his eyes? You see he's got red eye, you know, that you get from a camera flash, in one but not the other. That's because the other eye is a prosthetic eye. So this is Jake right after he was born, 2 months premature, and he only had one eye, and he had his toes were all fused together, and the most serious problem was that he had several blockages in his intestine. And so when he was a day old, 2 days old, he had to have surgery to cut those blockages out and pull the intestines back and stitch them together. Now, mind you, this is a preemie. This is a 7th-month old baby. It's not even ready to be born yet. Incredibly small. That intestine couldn't be much bigger than a piece of spaghetti. There are several layers that have to be stitched together, and the surgeons did it. And I just can't say enough about how amazing that is that they were able to do the surgery on him. And here, you know, this is last year at his 8th grade graduation. He's doing great. Kid's sharp as a tack. Well, because this was such a unique situation, my sister and my brother-in-law, they wanted to find out, well, what do we have any explanation for this? And they got tested, and they're all carrying these alleles. And so it's very likely that it's these alleles for this MTHFR gene which caused these developmental abnormalities in my nephew Jake. And so because of that, when we found all this out was right about the time that my wife got pregnant with our daughter. And so because we knew that this was a possibility, I got tested, my wife got tested. Turns out I'm heterozygous for this allele. Which would make sense, you know, my sister's got 2 copies. It's very likely that we at least have 1 copy in common. My wife also has this allele, and then she has a different mutant allele, the A1298C. And so that one also causes a substitution, but the protein does not seem to be affected, right? So you see we got the gamut here. You've got the substitution that causes a change in the amino acid, which causes the enzyme to not work very well. And then you've got a different allele with a different substitution that also causes a substitution in the amino acids, but the protein is fine. So it's a great example of what we've been talking about. And so because of my nephew, we got screened for this, and so we saw that because of my wife's genetic makeup that we're at an elevated risk for this kind of disorder. And so we were able to take preventative measures during the pregnancy. You know, so my wife was on ridiculous levels of folic acid, and she had to give herself blood thinner shots in her abdomen every day for her entire pregnancy. Bless her heart. And we would not have done that if we had not been able to screen for this. And you know, Sophie was a healthy baby and is doing great right now. So it just goes to show you the potential benefits of this knowledge. Okay, so I just wanted to tell you some interesting stories, help kind of connect what we've been learning to maybe some things you've experienced in your life. So please let me know if you got any questions, and that's all I have for right now, so I will talk to you later. Have a good night.